Welcome to Fluent Daily, the English listening channel for advanced learners at B2 and C1 level. Every episode features a rich, flowing conversation in natural English on a topic that is genuinely worth thinking about: history, psychology, science, philosophy, and more. There are no simplified sentences here, no slow speech, no vocabulary drills. Just real English spoken naturally on ideas that matter. This is Fluent Daily. Let's get into it.
Today we have a great story, so let's get started right now. Have you ever looked in the mirror, maybe tilted your head just a fraction, and been caught totally off guard because your grandmother's nose is suddenly staring back at you?
Oh yeah, it's that weird shock of recognition.
Right. Or maybe you have this incredibly specific quirk, a bizarre sneeze or a weird way of processing numbers or even a certain cadence in your laugh that just stubbornly echoes through your family tree generation after generation.
Which is fascinating because for the vast majority of human existence, that phenomenon was essentially magic. I mean, it was a complete black box.
Yeah, totally a black box.
We could see the inputs and the outputs. We knew, tall parents tended to have tall children. But the actual mechanism operating in the dark between those generations was a total void.
And today, we are tearing apart this incredible 170-year scientific thriller of how humanity finally wrenched that black box open. We're taking a deep dive into the source material that traces the shift from empirical pattern matching, like farmers just crossing the strongest horses and hoping for the best, to the discovery of the physical, mechanistic physics of life itself.
It really is a thriller.
It is. We were moving from a lonely monk, isolating traits in a garden, all the way to the, terrifying and awe-inspiring modern reality, where we hold the actual biochemical tools to actively edit our own species.
Okay, let's unpack this.
Well, the transition you're talking about from blind husbandry to molecular engineering, it represents arguably the most profound paradigm shift in the history of science. I mean, it rivals Newton codifying the laws of motion or Einstein bending space-time.
That's a massive comparison, but it fits.
It does, because this isn't just about categorizing plants and animals. It is the discovery of the underlying biological information system. It's the realization that life is fundamentally written in code. Right. And to find the person who first suspected that a code even existed, we actually have to travel back to the mid-19th century. So between 1856 and 1863, an Augustinian friar named Gregor Mendel is working in the garden of the St. Thomas Monastery in Brno.
Just a guy and his plants.
Exactly. But what sets Mendel apart isn't just that he's gardening, it's his background. He had trained in physics and mathematics at the University of Vienna under Christian Doppler. Yes, the Doppler effect guy.
Which completely changes how he approaches biology.
It changes everything. So Mendel doesn't look at plants like a traditional botanist. He looks at them like a statistician who's analyzing a physical system. And right from the start, his experimental design is just a masterpiece. He chooses the common pea plant.
And he chooses it because it solves the biggest problem in heredity research, which is random contamination.
Right, because pea plants are naturally self-fertilizing.
Exactly. The petals of the flower actually seal the reproductive organs tightly inside. So that anatomical quirk allowed Mendel to maintain absolute, dictatorial control over his data.
No random bees messing up the experiment.
None at all. He could open those immature buds, carefully snip away the pollen-producing anthers to prevent self-fertilization, and then take a delicate paintbrush and introduce pollen from one specific documented plant of his choosing. And he performed this microscopic surgery on tens of thousands of flowers.
Tens of thousands. It's mind-boggling patience. But the real genius, and honestly the terrifying stroke of sheer biological luck here, was in the specific traits he chose to measure. He looked at seed color, seed shape, pod color, stem height.
And that luck, it really cannot be overstated. When we look at the genetics of pea plants today, we know that the trace Mendel selected happened to be controlled by genes located on entirely different chromosomes, or, at least far enough apart on the same chromosome that they behave independently.
Wait, let me stop you there, because that is a massive point. What happens if, say, his brush slips, or he just gets curious, and he decides to track two traits that sit right next to each other on the exact same chromosome?
Well, the entire history of genetics probably stalls out for another 50 years.
Seriously? Just from picking the wrong trait?
Yeah, because if genes are physically tethered together on the same chromosome, a phenomenon we now call genetic linkage, they get inherited as a package deal. So if Mendel had chosen linked traits, say a specific petal color that was physically bound to a specific seed shape, His data would have shown those traits traveling together in weird, inseparable clumps.
So the math wouldn't work.
The beautiful, clean mathematical ratios he discovered, like the famous 3-to-1 or 9-to-3-to-1 ratios, they would have been distorted into incomprehensible noise. He would have looked at his ledgers and seen chaos instead of the foundational laws of heredity.
Wow, he dodged a massive bullet. Because he chose unlinked traits, he was able to formulate the law of independent assortment. Basically, he proved that inheriting a green pod has absolutely zero influence on whether that plant inherits a tall stem. The traits are processed as separate, independent variables.
Which naturally leads to his most revolutionary conceptual leap, right? To make his math work, Mendel had to invent a concept that flew in the face of every respected scientist of the 1860s. He proposed the existence of analogous, discrete, indivisible units of heredity.
We call them genes now, but at the time the whole world believed in blending inheritance, right?
Right. The idea was that biological traits were like liquids. If a tall person and a short person have a kid, their essences mix like pouring white paint and red paint into a bucket to get pink.
Yeah, the paint bucket theory. But the fatal flaw there, which Darwin himself struggled with immensely, is that if traits constantly blend, genetic variation would rapidly vanish.
Exactly. Within a few dozen generations, every individual and population would average out into an identical, uniform gray blob. Natural selection would have absolutely nothing to select from.
And Mendel's data totally annihilated the paint bucket theory. He proved that heredity is not a liquid. It's like a bag of marbles.
Bag of marbles, yes.
If you have a green pea marble and a yellow pea marble, they don't smear together to make a yellowish green marble. They remain entirely solid, discrete units that are shuffled and passed down intact. He completely solves the crisis of variation, and he writes up this earth-shattering revelation in his 1866 paper, Experiments on Plant Hybridization, and presents it to the world.
And the world responds with absolute deafening silence.
It's so sad.
It is one of the most agonizing ironies in the scientific canon. Mendel held the encryption key to biological life, and the scientific establishment completely ignored him for 34 years.
It wasn't until 1900 that 3 different scientists, Hugo de Vries, Carl Correns, and Erich von Schermach, were independently hitting the same mathematical conclusions. They dig through the archives, find Mendel's dusty paper, and realize this obscure friar had lapped them before they were even born.
Yeah, he beat them to the punch by decades.
So the turn of the 20th century gives us the math. We know these discrete units exist. But a mathematical unit is essentially a ghost. If this information is being physically transported from a parent to a child, it has to have a shape, it has to have mass, it has to be a molecule.
Which sets off a massive biochemical manhunt.
Right.
For decades, the prime suspects were actually proteins, because chromosomes inside the nucleus are made of 2 things, proteins and nucleic acids. And proteins are incredibly complex, right? They're built from 20 different amino acids capable of folding into infinite shapes.
They seem like the obvious choice for a complex code.
Exactly. Meanwhile, deoxyribonucleic acid, or DNA, is built from just 4 simple chemical bases. To a 1930s biochemist, assuming DNA carried the complex instructions for a human being was like assuming you could write a Shakespearean play using a 4-letter alphabet. It just seemed too stupidly simple.
Until a series of experiments in the 40s and early 50s proved definitively that it actually is the DNA carrying the data. And suddenly, every lab in the world pivots.
The new obsession becomes, what does this molecule actually look like?
Because if we can see its shape, maybe we can understand how it stores information, which initiates, probably the most dramatic cutthroat race in modern scientific history.
Oh, absolutely. And the clash of methodologies here is fascinating. Over at Caltech, you have Linus Pauling, who is the undisputed heavyweight champion of structural chemistry. Pauling's approach is highly theoretical and model-based. He had already cracked the alpha helix structure of proteins.
But Pauling makes a catastrophic, unforced error. He proposes a triple helix model for DNA, but he rushes it, right? He places the negatively charged phosphate groups on the inside of the molecule, clustered together in the core.
Which is wild, because any first-year chemistry student can tell you why that fails. Like charges repel. If you pack negatively charged phosphates into the center of a molecule without neutralizing them, the electrostatic repulsion would cause the entire structure to violently dough itself apart.
It's physically impossible.
Complete.
So while Pauling is publishing this chemically impossible model, you have 2 camps in England closing in on the truth. Over at Cambridge, James Watson and Francis Crick are playing with metal and cardboard cutouts, trying to piece the molecule together like a jigsaw puzzle, based on other people's data.
And at King's College London, Rosalind Franklin and Maurice Wilkins are generating that actual data. They are using X-ray crystallography, which, I have to emphasize, is an intensely difficult technique.
Yeah, it's not just taking a picture through a microscope.
Not at all. You have to extract the DNA, draw it into a microscopic fiber where all the molecules align perfectly, and then bombard it with X-rays. The X-rays bounce off the atoms in the DNA and scatter onto a photographic plate, creating a diffraction pattern.
We have to pause on the physics of this for a second because understanding the mechanism of crystallography is the only way to understand what happens next. The image on the photographic plate doesn't look like a spiral staircase. It's not a photograph of a molecule. It's basically a scatter plot of dots.
Right, and the math translating that scatter plot into a 3D structure is brutal. But there are signatures. When X-rays hit a helical structure, they diffract in a very specific recognizable pattern. It's an X shape. The angle of the X tells you the pitch of the helix. The spacing of the layer lines tells you the distance between the turns.
And Rosalind Franklin was an absolute master of this. She had refined her technique to capture an image of the hydrated B form of DNA. This is the legendary photo 51.
The most famous photo in biology.
It really is. It is a stunningly crisp image showing a massive, undeniable X. It doesn't just suggest a helix, it mathematically demands a helix, and it provides the exact physical dimensions of its diameter and twists.
Which brings us to the profound ethical fracture at the heart of the discovery. Maurice Wilkins and Rosalind Franklin had a notoriously toxic working relationship.
Yeah, they really did not get along.
No. And without Franklin's knowledge and completely outside the bounds of professional consent, Wilkins takes photo 51 and shows it to James Watson.
Which is just, it's such a betrayal, and Watson himself later writes that the moment he saw the photo, his mouth fell open and his pulse began to race. He didn't even need to do the complex Fourier transform math. Just looking at the clean cross of that X, he knew it was a helix.
Furthermore, Franklin's previous reports, which also ended up in Watson and Crick's hands, proved that the phosphates had to be on the outside of the molecule, interacting with water. She had already solved the error that doomed Linus Pauling.
So armed with Franklin's proprietary data and structural parameters, Watson and Crick returned to their cardboard cutouts, and they realized the only way the structure fits together is as a double helix 2 anti-parallel strands running in opposite directions, with the phosphates forming a structural backbone on the outside.
Right, and on the inside, bridging the gap between the two strands are the chemical bases.
Yeah.
Adenine, thymine, cytosine, and guanine A, T, C, and G. Watson discovers that if he pairs an A with a T and a C with a G, the physical width of those pairs is identical. They form perfect, uniform rungs on the spiral staircase.
It's so elegant.
What's fascinating here is that the brilliance of the 1953 Watson and Crick model, published in Nature, is that the structure instantly answers the mystery of heredity. The shape of the molecule is the copying mechanism.
Because of the hydrogen bonds, I love this part, the backbone of the DNA strand is held together by incredibly strong covalent bonds. But the rungs of the latter, the AT and CG pairs, are held together by relatively weak hydrogen bonds. It's basically like a molecular zipper.
Exactly. So if you need to copy the genetic code, an enzyme simply runs down the middle of the double helix, breaking those weak hydrogen bonds, and unzips the two strands.
Just unzips it down the middle.
Right. And because A only ever pairs with T, and C only ever pairs with G, each single strand serves as a perfect foolproof template. You float new chemical letters in, they lock into their complementary partners, and suddenly you have two identical double helices where you used to have one. The mechanism of replication is built directly into the chemistry.
It is the most beautiful molecule in the universe. But the legacy of how we found it remains deeply, deeply contested. Watson, Crick, and Wilkins share the 1962 Nobel Prize. Rosalind Franklin had died of ovarian cancer 4 years prior at the age of 37.
Yeah, and the Nobel rules strictly forbid posthumous awards, which is the administrative reason she wasn't on the stage in Stockholm. But the historical reality is much darker.
Much darker.
The systemic minimizing of her contribution in the initial publications, compounded by Watson's patronizing portrayal of her in his later memoir, The Double Helix, it really reveals the pervasive gendered hostility of the era. I mean, her experimental brilliance provided the physical scaffolding that allowed the theoretical leap to happen.
And acknowledging that isn't just about settling historical scores, you know? It forces us to examine the culture of science itself and whose labor is classified as support versus whose is classified as genius. But scientifically, the discovery of the double helix unleashes a new, even more maddening puzzle. We know the shape. We know how it copies itself, but knowing the shape of a hard drive doesn't tell you how the software renders a video on your screen. How does the sequence of A's, C's, T's, and G's trapped inside a cell nucleus actually construct the physical tissues of a human body?
And solving that question became the defining project of the 1960s, culminating in the framework Francis Crick termed the central dogma of molecular biology.
The central dogma.
Yes. It posits a strictly one-way flow of biological information. DNA is transcribed into RNA, which is then translated into protein.
I want to dig deeply into the mechanism of this translation, because this is where inorganic chemistry basically turns into living biology. Scientists like Marshall Nierenberg and Hargo bin Khorana set out to crack the cipher. The body runs on proteins, enzymes, muscle fibers, antibodies, and proteins are long chains assembled from 20 different kinds of amino acids.
But the mathematical hurdle was obvious. DNA only has 4 letters. If one DNA letter coded for one amino acid, you could only specify 4 amino acids.
Which is nowhere near the 20 we need.
Right. If you read the letters in pairs, 4 squared gives you 16 combinations. Still not enough.
So it has to be triplet code. 4 to the power of 3 gives you 64 possible combinations.
Exactly, 64 possible 3-letter sequences, which we call codons. But how do you prove which 3-letter codon matches which specific amino acid?
Nierenberg's approach to this was absolute genius. You can't see this happening in a microscope, so he builds a synthetic system, the famous poly-U experiment.
Oh, this is a classic.
Yeah, it really is. He synthesizes an artificial strand of RNA consisting exclusively of the letter uracil, U, just repeating forever.
Yeah.
He drops this synthetic RNA into a test tube filled with ribosomes and all 20 amino acids. He lets the cellular machinery run, and he analyzes the protein it spits out.
And the resulting protein was a chain made entirely of just one amino acid, phenylalanine.
Boom. UU equals phenylalanine. He essentially created the Rosetta Stone.
But through painstaking biochemical assays, they eventually map all 64 codons. And they discover a brilliant evolutionary safeguard built into the math.
What's the safeguard?
Well, if we have 64 codons, but only 20 amino acids to assign them to, we have a surplus, right? 3 of those codons act as structural stop signs, telling the ribosome to terminate the protein chain. The remaining 61 all code for amino acids.
Meaning the code is degenerate. Multiple different triplets code for the exact same amino acid.
Right. This phenomenon, often driven by what Francis Crick called wobble in the third position of the codon, is a profound buffer against mutations. If UV radiation or replication error accidentally swaps the third letter of a codon from a C to a G, the cellular machinery often still grabs the exact same amino acid. The protein folds perfectly. The organism survives unharmed.
That is amazing. Let's visualize this entire flow of information, because picturing the scale of this machinery happening inside your cells right now is mind-bending. I always picture the nucleus of the cell as like a heavily fortified ancient monastery vault.
I like that. So the DNA is the master text locked inside that vault.
Exactly. It is much too valuable, much too prone to degradation to ever be allowed out into the chaotic main floor of the cell. If a protein needs to be built, a specialized enzyme RNA polymerase acts as a master scribe. It enters the vault, unwinds a tiny specific section of the master text, and transcribes those DNA letters into a cheap, single-stranded, temporary scroll. That scroll is messenger RNA, or mRNA.
And once the mRNA scroll is transcribed, it gets heavily edited and then exported through the pores of the nuclear vault out into the cytoplasm.
It goes to the monastery's kitchen. And waiting in the kitchen is the ribosome, acting as the head chef. The ribosome clamps onto the mRNA scroll and starts reading the letters in groups of 3, the codons. For every codon it reads, smaller molecules called transfer RNAs, or tRNAs, act as the prep cook.
The prep cook's running around.
Yeah. They scramble into the pantry, find the single specific amino acid that matches that 3-letter codon, and drag it back to the chef. The ribosome snaps that amino acid into a growing chain, shifts exactly 3 letters down the scroll, and calls for the next ingredient.
And it does this millions of times a second, continuously, in nearly every cell of your body, chaining these amino acids together until they fold into a 3-dimensional protein capable of forming a hemoglobin molecule or retinal receptor in your eye.
It's just wild to think about. And the most humbling realization to come out of Nierenberg and Khorana's dictionary is its universality. The specific codon that tells your human ribosomes to add the amino acid methionine is the exact same codon doing the exact same job in the cells of a blue whale, an oak tree, and a microscopic deep-sea bacterium.
Yeah, aside from a few microscopic variations in certain mitochondria, the genetic code is ubiquitous. It strongly implies that this specific translational machinery evolved once, billions of years ago, in a single ancestral population, and was passed down unbroken to every living thing on this planet. It is the molecular proof of our shared biological heritage.
Knowing this language inevitably triggered the most ambitious mapping expedition in human history. Because if we have the dictionary, the obvious next step is to sit down and read the entire instruction manual.
Enter the Human Genome Project. Officially launched in 1990, it aimed to sequence all 3 billion base pairs of the human genome.
3 billion letters. To grasp the sheer physical magnitude of that data, if you were to print the human genome into standard paperback books, it would fill a library of over 4,000 volumes. And the technology in the 1990s was painfully slow.
It was. The publicly funded international consortium was using a highly methodical approach called hierarchical shotgun sequencing.
How did that work?
They essentially built a map first. They cut the genome into large, manageable chunks, cloned those chunks into bacteria, figured out roughly where each chunk belonged on the chromosomes, and then slowly sequenced the individual letters within those chunks. It was precise, deeply organized, and glacially slow.
Which created a massive opening for disruption. In 1998, a private company called Celera Genomics, led by Craig Venter, announced they were going to sequence the genome faster, cheaper, and entirely privately.
Yeah, and Venter employed a radically different strategy known as whole genome shotgun sequencing. He bypassed the meticulous mapping phase entirely. Instead, he took the entire 3 billion letter genome and subjected it to high-frequency sound waves, violently shattering it into millions of tiny, random, overlapping fragments.
He just blew the whole book up into sentence fragments.
Exactly. Then he sequenced all those tiny fragments simultaneously. The challenge wasn't the biochemistry, it was the computer science. Celera had to write entirely new algorithms and utilize some of the most powerful supercomputers on the planet to analyze these millions of fragmented strings of text, look for overlapping sequences at the edges, and mathematically stitch the 3 billion letters back together in the correct order.
But the public consortium argued that Venter's computational approach would catastrophically fail, right? Because the human genome is full of highly repetitive sequences.
That was the great fear. Our genome is littered with elements like ALU sequences, stretches of a few hundred letters that repeat identically thousands of times. If you blow the genome apart and you have thousands of identical puzzle pieces, an algorithm can easily get lost trying to reassemble them, causing major sections of the genome to collapse in the computer model.
But the threat of Celera privatizing and patenting the human genome forced the public consortium into overdrive. They wildly accelerated their own sequencing machines. The race became a massive geopolitical event, culminating in 2000 with a joint announcement at the White House declaring a tie. And by 2003, they had published the essentially complete reference genome.
And it fundamentally rewrote our understanding of our own complexity. Prior to the project, estimates suggested humans might have up to 100,000 protein-coding genes. We assume that because we are behaviorally and neurologically complex, our parts list must be massive.
And the final tally comes in at roughly 20,000 genes.
Right. We have fewer protein-coding genes than a tiny water flea and roughly the same number as a microscopic nematode worm.
It's almost insulting.
It kind of is. But it forced molecular biologists to realize that human complexity doesn't come from the raw number of genes. It comes from how those genes are regulated, how they're spliced together in multiple different combinations, and how the non-coding regions of the DNA orchestrate the timing and expression of those 20,000 parts.
Here's where it gets really interesting, because this realization collided violently with the public hype of the early 2000s. The media promised us that because we had the blueprint, we were going to find the gene for everything. The gene for intelligence, the gene for schizophrenia, the gene for being a great piano player.
And that era of genetic determinism has been thoroughly dismantled by the advent of genome-wide association studies, or GWAS.
Right, because once sequencing became incredibly cheap, we didn't just sequence one reference genome. We started sequencing hundreds of thousands of people, overlaying their health data, and running massive statistical regressions to see which variations in the code correlated with which diseases.
And GWAS proved conclusively that complex traits are highly polygenic. There is no single gene for depression or height. Instead, traits are the aggregate result of hundreds, sometimes thousands, of single-letter variations scattered across the entire genome.
These are the polygenic risk scores we hear about.
Yes. Each individual variation might increase your risk of a disease by, like, 0.01%. Furthermore, we discovered the pervasive reality of pleiotropy, which is that a single gene doesn't just do one thing. It might affect your immune system, your brain chemistry, and your bone density simultaneously. You cannot isolate complex human biology into simple, modular genetic switches.
But we shouldn't let the failure of the single gene myth overshadow the actual staggering triumphs of genomic medicine. The ability to read the code has totally revolutionized fields like pharmacogenomics.
Oh, absolutely. Pharmacogenomics fundamentally shifts medicine from a one-size-fits-all approach to molecularly tailored therapies. We now know that specific genetic variations dictate the exact shape of the enzymes your liver uses to metabolize drugs.
So the CYP2D6 gene is the classic example here, right?
It is critical. The CYP2D6 enzyme processes a huge percentage of common medications, including opioids like codeine. Codeine itself is actually a prodrug. It does nothing until your liver enzymes convert it into active morphine. If your genome contains duplicated copies of the CYP2D6 gene, you are what we call an ultra-rapid metabolizer.
Which sounds like a good thing, but it's actually a nightmare.
It can be lethal. If a surgeon gives an ultra-rapid metabolizer a standard, totally routine dose of codeine after having their tonsils removed, their hyperactive liver enzymes will instantly dump a massive, overwhelming dose of morphine into their bloodstream. They can suffer fatal respiratory depression from a completely standard prescription.
Wow. And on the flip side, if you are a poor metabolizer, your liver can't convert the codeine at all, and you are left in agonizing post-op pain while the doctors accuse you of faking it. By simply sequencing that one gene, we can prevent lethal toxicity and guarantee therapeutic efficacy.
We see the exact same critical application in oncology. Cancer is not a single disease of an organ. It is a disease of the genome. It is caused by acquired mutations that break the cellular mechanisms controlling growth and division.
So today, an oncologist doesn't just look at a lung tumor under a microscope. They extract the DNA from the tumor cells themselves and sequence it. They look for the exact typographical error driving the uncontrollable growth.
Exactly. And if they find a specific mutation, like an EGFR mutation, they can deploy a targeted molecular inhibitor that binds exclusively to that mutant protein. It shuts down the cancer at the biochemical level while largely sparing healthy tissue. The genome has basically become the ultimate diagnostic tool.
So we learned the alphabet. We deduced the structure. We deciphered the translation dictionary. And we read the entire 3 billion letter manual. But human beings are fundamentally engineers, you know. The moment you hand us a completed manuscript, we immediately start looking for a pen and a bottle of whiteout.
Which introduces the technology that promises and threatens to redefine the trajectory of life on Earth, CRISPR-Cas9.
Developed by Jennifer Doudna and Emmanuel Charpentier, a breakthrough that earned them the Nobel Prize in Chemistry in 2020. It's a programmable genetic word processor.
To appreciate the elegance of CRISPR, you have to understand that humans did not build it from scratch in a chemistry lab. We hijacked a system that has been undergoing billions of years of evolutionary beta testing in bacteria. CRISPR is a natural adaptive immune system.
It's how bacteria fight off viruses.
When a bacteriophage virus injects its lethal DNA into a bacterium, if the bacterium manages to survive, it does something extraordinary. It snips out a tiny fragment of the attacking virus's DNA and physically inserts it into its own bacterial genome, into a region characterized by clustered, regularly interspaced, short palindromic repeats, which is where we get the acronym CRISPR.
It basically takes a mugshot of the invader and archives it in its own hard drive.
That's a great way to put it. If that exact strain of virus ever attacks again, the bacterium transcribes that archived mugshot into a small piece of guide RNA. It attaches this guide RNA to a large DNA-cutting protein enzyme called Cas9.
So this molecular assassin complex patrols the inside of the cell. The guide RNA is constantly checking the sequence of any foreign DNA it bumps into. And if the RNA perfectly matches the viral DNA,
the Cas9 protein undergoes a rapid conformational change. It physically locks onto the viral DNA, unzips it, and its enzymatic domains cleave both strands of the double helix cleanly in half, instantly destroying the virus.
What Doudna and Charpentier realized is that you can swap out the mugshot. You can synthesize your own custom guide RNA in a lab, matching any sequence of letters you want, and attach it to Cas9.
You can inject this complex into a human cell. The guide RNA will navigate through the dense, chaotic soup of the nucleus, scan past billions of base pairs, find the one specific 20-letter sequence you programmed it to find, and Cas9 will execute a precise double-stranded cut at that exact location.
But just cutting the DNA isn't editing it, right? The edit happens in the panic that follows.
Right, because a double-stranded DNA break is a lethal emergency for a cell. The cell's internal repair machinery scrambles to stitch the severed ends back together. If you simply let the cell attempt to fuse the ends, a process called non-homologous end joining, it usually makes mistakes. It adds or deletes random letters, which scrambles the codon reading frame and effectively disables the gene entirely. We call this a gene knockout.
You've turned off a malfunctioning system, but if you want to actively rewrite the code, you supply a template.
Exactly. Alongside the CRISPR machinery, you flood the nucleus with a custom-designed strand of DNA containing the sequence you want to be there. The cell, desperately looking for a way to bridge the gap smoothly, will use a process called homology-directed repair. It copies the sequence from your template and patches it seamlessly into the genome. You have just replaced a disease-causing mutation with a healthy sequence.
We had older gene editing tools before CRISPR, like zinc finger nucleases and TALENs, but they were incredibly expensive, finicky, and required engineering massive custom proteins for every single edit.
CRISPR democratized genetic engineering because the cutting protein Cas9 never changes. The only thing you have to alter to target a new gene is the tiny, incredibly cheap strand of guide RNA. You can order custom guide RNA on the internet for less than the cost of a textbook. Within a few years, researchers went from struggling to edit single cells to routinely engineering the genomes of mice, crops, and primates.
And now, human beings. The translation into clinical medicine has been terrifyingly fast. In 2023, regulatory agencies around the world, including the FDA, approved the first CRISPR-based therapeutic Casgevy for sickle cell disease.
Sickle cell is a devastating monogenic disease. A single-letter mutation in the gene coding for beta-globin causes hemoglobin proteins to stick together in long, rigid chains. This forces the red blood cells to warp into a sharp sickle shape. They clog capillaries, causing catastrophic oxygen deprivation, agonizing pain crises, and severe organ damage.
And the CRISPR cure is just a triumph of molecular engineering.
Doctors extract hematopoietic stem cells from the patient's bone marrow. In a sterile manufacturing facility, they use CRISPR to edit the DNA within those cells. Specifically, they cut a regulatory region to force the cells to start producing fetal hemoglobin, a type of hemoglobin the body normally stops making after birth, which is completely unaffected by the sickle mutation.
They obliterate the patient's remaining diseased bone marrow with chemotherapy and then infuse the freshly edited, supercharged stem cells back into the body. The cells engraft, start pumping out healthy, round, red blood cells, and the patient is functionally cured at the DNA level.
It is a miracle of modern science. But, it also highlights the limitations. The treatment is brutally grueling for the patient, requires highly specialized infrastructure, and carries a price tag in the millions of dollars. Scaling this technology to the millions of people suffering from genetic diseases globally remains an unsolved socioeconomic crisis.
But the power to alter human DNA forces a reckoning with a much darker, much more dangerous boundary. In 2018, a biophysics researcher in China named He Jiankui crossed a red line that the entire international scientific community had agreed not to cross.
Yes, he announced that he had successfully utilized CRISPR to edit the genomes of viable human embryos. Those embryos were subsequently implanted, leading to the birth of twin girls, the first gene-edited human beings in history.
The global reaction wasn't just concern, it was visceral, unified horror. What exactly did he edit, and why was it such a catastrophic violation?
He targeted a gene called CCR5. The CCR5 protein sits on the surface of certain immune cells and acts as a primary co-receptor, essentially a locked door that the HIV virus utilizes to infiltrate the cell. By using CRISPR to knock out this gene and disable the receptor, his stated objective was to engineer children who possessed innate genetic resistance to HIV infection.
I mean, on the surface, if you don't know the biology, engineering HIV immunity sounds like a noble medical goal. But when you unpack the details, the entire experiment collapses into a nightmare of unethical conduct.
The condemnation centered on several profound failures. First, the risk-to-benefit ratio was entirely unjustifiable. We already have robust, highly effective, safe protocols for preventing the transmission of HIV from an infected parent to a child. Exposing an embryo to an experimental, unproven, potentially mutagenic genome editing procedure to solve a problem that already has a safe solution is a grievous breach of medical ethics.
He also performed the work in secret, forging ethical review documents and evading proper institutional oversight.
But the core reason this incident galvanized global panic is the fundamental biological nature of the edit itself. He, Jiankui, did not perform somatic gene therapy. He performed germline gene editing.
This is the ultimate ethical fault line in modern genetics. We have to clearly define these two categories because the future of the human race pivots on the distinction.
Somatic cells are the fully differentiated cells that make up your body, your liver, your skin, your lungs, your blood. When doctors treat a sickle cell patient with Casgevy, they are editing somatic stem cells. Those edits are confined exclusively to that individual patient. Crucially, they are not passed on to the patient's children. If the patient dies, the edited DNA dies with them.
So somatic editing is fundamentally just a highly advanced pharmaceutical. It treats an existing, consenting patient.
Germline editing, conversely, involves altering the DNA of gametes, sperm, or eggs, or early-stage embryos. When Jian Kui edited those embryos at the single-cell stage, the CRISPR-altered DNA was copied into every single subsequent cell as the embryo developed.
Including the cells that will eventually become the girl's own eggs.
Precisely. A germline edit is heritable. The deletion in the CCR5 gene will be passed down to their children, their grandchildren, and every descendant that follows. He did not just alter two patients. He permanently introduced a synthetic genetic alteration into the collective human gene pool.
He made a decision for hundreds of future generations who could never consent to the risks.
Which forces us into the most complex bioethical debate of the century. There is a developing cautious consensus among international scientific bodies that germline editing might one day be morally justifiable, but only under incredibly strict conditions. Specifically, restricting it solely to the prevention of catastrophic, lethal monogenic diseases where no other alternative exists, like Huntington's disease or Tay-Sachs.
But the moment we master the technology to safely edit out Huntington's disease, we inherently possess the exact same technology to edit in traits. This is the debate over genetic enhancement.
Designing human beings to possess traits that exceed the normal, natural human baseline. Utilizing CRISPR to engineer higher cognitive capacity, enhanced muscular density, greater memory retention, or specific cosmetic phenotypes like eye and hair color.
I'm going to aggressively play devil's advocate here, because the arguments against enhancement often rely on a romanticized view of natural human biology that we don't apply to any other area of life. Bioethicists like Julian Savulescu make a compelling, highly provocative argument for the moral imperative of enhancement.
It's an uncomfortable argument for many, but it's logically rigorous.
It is. Consider the reality of human behavior. Parents currently spend immense sums of money and energy actively enhancing their children. We optimize their diets with specific synthetic vitamins. We inject them with vaccines to train their immune systems. We subject them to rigorous, expensive educational environments to physically alter the neural pathways in their brains, attempting to increase their intelligence. We consider all of this environmental manipulation to be the definition of excellent parenting.
We do.
So, if we accept that maximizing a child's potential is a moral good, and if a genetic edit achieves the exact same outcome as 10 years of expensive tutoring, but does it more effectively, why is the genetic edit arbitrarily classified as a moral violation? If we possess the technical capability to grant a child a robust biological baseline resistance to Alzheimer's, a stronger cardiovascular system, a more resilient neurochemistry, doesn't refusing to use that technology constitute a form of medical neglect? Why should we leave the architecture of a human being to the brutal, unthinking, chaotic lottery of natural Darwinian selection when we can apply rational, compassionate design?
The argument for procreative beneficence, the idea that we are obligated to select the best possible genetic starting point for our offspring, is logically powerful, precisely because it highlights how heavily we already intervene in natural processes. However, the counter-arguments from the broader bioethics community expose the sheer, terrifying complexity of treating the human genome like a software program we can casually upgrade.
The risks are massive.
Huge. Firstly, the biological reality of pleiotropy, which we discussed earlier, makes enhancement incredibly dangerous. You cannot simply tweak a gene to increase memory retention without almost certainly causing cascading, unpredictable effects across other neurological systems. The genome is a delicately balanced ecosystem tuned by millions of years of evolutionary trade-offs.
You try to edit in higher IQ and accidentally bake in severe chronic anxiety.
Exactly. Secondly, the societal implications are dystopian. Genetic enhancement technologies will inevitably be astronomically expensive. If only the socioeconomic elite can afford to engineer biological advantages into their children, we will literally encode wealth inequality into the human genome. We risk speciating humanity into a genetically optimized ruling class and an unenhanced underclass.
That's a terrifying thought.
Furthermore, there is a profound philosophical shift in how we view the relationship between parent and child. Currently, a child is viewed as a gift, an independent biological entity with their own inherent unscripted potential. If we pivot to a model of genetic enhancement, the child becomes a product. a manufactured commodity designed to fulfill the specific aesthetic or functional desires of the parents. This commodification fundamentally erodes the unconditional acceptance that underpins the parental bond.
We shift from accepting the child we receive to demanding the product we engineered.
And from an evolutionary standpoint, aggressive enhancement risks devastating the genetic diversity of our species. If society collectively decides that a specific set of traits, say, tall stature, extroversion, and mathematical proficiency, are universally desirable, and we genetically select for them, we drastically narrow the human gene pool based on the fleeting cultural biases of a single century. Evolutionary survival is entirely dependent on maintaining a vast reservoir of genetic diversity to adapt to unforeseen future challenges. Homogenizing the human genome is an existential threat.
The staggering thing is that these debates are no longer confined to academic symposiums. The technology has matured so rapidly that it has permeated our culture, our media, and our legal frameworks.
The vocabulary of the molecular age is now a prerequisite for basic civic engagement. You cannot understand debates regarding health insurance discrimination, forensic data privacy, or pharmaceutical pricing without a functional literacy in concepts like DNA, RNA, mutation, heritability, and genome sequencing.
So what does this all mean for daily life?
Well, if we connect this to the bigger picture, it requires us to confront the most pervasive, dangerous misunderstanding propagated by popular media regarding how genetics actually works. To navigate this information landscape, you must rigidly maintain the conceptual distinction between genotype and phenotype.
The genotype is the raw data. It is the literal sequence of 3 billion letters, A, C, T, G, sitting inside your nucleus.
Yes, your genotype is the static blueprint. Your phenotype, however, is the physical reality. It is your observable characteristics, your height, your resting heart rate, your immune response, your behavioral tendencies. The media consistently conflates the two. They produce headlines claiming scientists have discovered the gene for violence or the gene for alcoholism. This collapsing of genotype and phenotype fuels the insidious philosophy of genetic determinism.
Genetic determinism is the idea that your DNA is an inescapable script, that if you have the bad variant of a gene, you are mechanically destined to develop the disease or the behavior, regardless of what you do.
And modern biology has proven that determinism is demonstrably, unequivocally false. Your phenotype is never a direct one-to-one translation of your genotype. Your phenotype is the continuously evolving product of your genotype colliding with your environment.
That collision is mediated by epigenetics, right?
Yes. Environmental factors, your diet, your exposure to stress hormones in utero, the trauma you experience, the toxins in your air, the quality of your sleep, physically interact with your genome. They don't change the sequence of the letters, but they trigger biochemical modifications like DNA methylation or histone acetylation, which act as physical volume knobs on your genes. The environment dictates which genes are turned on, which are aggressively amplified, and which are completely silenced.
So your genome is the keyboard, but your environment is the musician playing the song.
If we surrender to genetic determinism, we abdicate our social responsibility. Why fight for clean air, equitable education, or robust social safety nets if human outcomes are simply preordained by the DNA? The reality is that the human genome provides the initial parameters, but the profound complexity of environmental interaction authors the final human being.
We have covered a monumental expanse of scientific history today. We started in a quiet monastery garden in Brno, where Gregor Mendel proved that the physical traits of living things are passed down in discrete, indestructible packets of information.
We traced the biochemical manhunt that proved those packets were composed of DNA, leading to the breathtaking discovery of the double helix by Watson, Crick, and Rosalind Franklin, revealing how the molecular structure itself enables the copying of life.
We watched Nirenberg and Karana decipher the universal triplet code, proving that a bacterium and a human being speak the exact same molecular language to translate DNA into the proteins that build our bodies.
We saw the brute force computational triumph of the Human Genome Project, successfully mapping all 3 billion letters of our own blueprint, and the subsequent realization through GWAS that our biology is infinitely more complex and interconnected than we ever imagined.
And we arrived at the precipice of the modern era, where CRISPR-Cas9 grants us the godlike ability to rewrite the code, curing devastating genetic diseases, while simultaneously forcing us to navigate the ethical minefield of germline editing and human enhancement.
This 170-year journey is not just a timeline of biological discoveries. It is the story of humanity discovering its own source code And I urge you to remember that your own genome, the DNA physically coiled inside the nucleus of the cells in your body right now as you listen to this, is a 3 billion-letter historical document. It is operating via the exact same molecular translation machinery we discussed, a continuous, unbroken chain of molecular information connecting you to the very origin of life on Earth.
But before we sign off, I want to leave you with one final, deeply provocative thought experiment that builds directly on the crucial distinction between genotype and phenotype we just established. We just explored how your phenotype, your physical reality, is the result of your static genotype dynamically interacting with your environment through epigenetic triggers. Well, what happens when humanity's environment undergoes a radical, unprecedented shift?
Like space exploration.
Exactly. Think about the next few centuries. As we establish permanent colonies in deep space, on the moon, or under the crushing gravity and irradiated atmosphere of Mars, or even if we remain on Earth, but our climate drastically transforms the atmospheric composition and temperature of the planet.
The environmental inputs would be entirely alien to mammalian biology.
Right. Our bodies will be subjected to levels of cosmic radiation, altered gravitational loads, synthetic diets, and atmospheric pressures that human DNA has never encountered in its 300,000-year evolutionary history. Without scientists deliberately altering a single letter of our genetic code without touching a single CRISPR molecule, our unedited, historically identical human genotypes will suddenly be forced to interact with wildly alien environments. The epigenetic response will be massive.
It would be unprecedented.
Could our physical human phenotypes shift and adapt so dramatically in response to those extreme environmental triggers that just a few generations down the line, we look, function, and behave like an entirely new species? Could we become unrecognizable to ourselves, all while possessing the exact same underlying human genotype we carry today?
It forces us to realize that human evolution moving forward might not be driven solely by rewriting the letters of the code, but by radically altering the context in which that code is expressed.
It is definitely something to think about the next time you look in the mirror and spot your grandmother's nose. The exact same genetic instructions she passed down to you might build something entirely different under the red sky of another world. Thank you for diving into the molecular machinery of life with us today.
It has been a profound exploration. Keep questioning the world around you and never stop examining the complexities within.
We will see you next time on The Deep Dive.